Hello everyone! Today we're going to be talking about the process of meiosis. So we know that humans have 46 chromosomes, right? So we have got 23 pairs. 23 comes from mom, right? So each of these that are in with a red box comes from mom, and 23 come from dad. So we see them paired here because these two chromosomes that are chromosome 1 from mom, 1 from dad, these have the same genes, but different types of genes, right? So it might be the gene for hair color, the gene for eye color, the gene for skin color, but you have mom's version of that gene and then dad's version of that gene. Okay, so when we say 23 pairs of chromosomes, this is what we mean. In the last pair, the X and Y determine if you're biologically male or female. But this process of meiosis is special because only sperm and egg are made this way. So we're going to learn today how sperm and egg are made. While all the other body cells divide by mitosis, these special cells have a different way of dividing so that each individual organism is different, right? We're not just clones of each other. Okay, so meiosis, a love story. How sex cells are made. So first let's back up a little bit and talk about the two types of eukaryotic cells that I just mentioned. First type is called somatic cells. So the somatic cells are the cells that we've already learned about. These are the body cells. They contain a diploid number of chromosomes. This just means a normal number of chromosomes, which is represented by 2n. So this means we have a pair, essentially, two chromosomes for each pair, right? So humans have 46. That's a 2n number. So some examples of somatic cells are basically any cell that's not a sperm or an egg. Skin cells, blood cells, brain cells, liver cells, muscle cells, bone cells, all of those other types of cells. And these divide by mitosis, which we've already learned about, right? So when a skin cell divides, it makes a copy of itself like a clone that's genetically identical to the original cell. Those are somatic cells, body cells. The other type of eukaryotic cell is a gamete. So these are the ones we're going to be learning about today. These are sex cells, so sperm and egg, and they only contain half the number of chromosomes or a haploid number. Hap kind of sounds like half to me. That's how I remember it. It's also represented by just the letter N, right? Where 2N is diploid, haploid is just 1N. So these examples would be sperm cells and ovum, which is egg cells, or ova is plural. They divide by meiosis, which is similar to mitosis, but we'll look more in depth with that in a minute. The male gametes, or the sperm cells, are produced in the male gonad, which is the testes. So gonad is just a word for the organ that's producing the sex cells. So the male version of that organ is called the testes. The female gonad is the ovaries. So we have the testes here, that's where sperm are produced in the male. So that's where this process of meiosis happens in males. And the ovaries, which are located right here, are producing the eggs in female, or the ova. Okay, so fertilization is the union of those two special cells. So when the sperm and egg come together, sperm plus egg, we get fertilization. So that's when we end up with a fertilized egg, which is called a zygote. Okay? So the zygote is a fertilized egg that has a diploid number of chromosomes. Okay, so for example, the sperm cell, sperm is N, haploid, right? The egg cell has a haploid number of chromosomes, right? And when they come together, they make a zygote, which is 2N, a diploid. So two haploid cells come together and their chromosomes unite, and we end up with twice the number of chromosomes, or a diploid number of chromosomes. Okay? So here's another visual of that happening. We've got the egg with 23 chromosomes, sperm with 23 chromosomes. When they come together, we end up with a zygote, 46 chromosomes. And this zygote is what develops into an embryo, eventually a fetus, and is born into a human or whatever organism it is. The same thing happens with animals and even plants. Okay, so now how are these special cells made, right? Because how come humans don't just all look like clones of each other, right? Or animals or plants, for example. We have variations, and it's all because of this amazing process of meiosis. So this is the process that produces haploid gametes, or sex cells. It's similar to mitosis, so we have kind of the same names of the phases, which you'll see, but this time it happens twice. So there's 2 rounds of division, and one diploid cell in the beginning ends up producing 4 haploid cells. So we see 4 haploid gametes here. So one diploid cell makes 4 sperm or 4 eggs. All right, so let's start in the beginning. Prophase 1. Right, remember I said it's the same phases of mitosis but twice. So the first 4 are prophase, metaphase, anaphase, telophase 1. The next 4 are prophase, metaphase, anaphase, telophase 2. So this is where duplicated homologous chromosomes. Homologous means same, right? Homo means same. So these are pairs of chromosomes that have the same genes like I talked about in the beginning. And they're replicated, right? So they look like X's. So we have mom, or paternal is dad. So dad's chromosome 1 and mom's chromosome 1. So those homologous chromosome pairs, so mom and dad's chromosomes, pair together to form something called a tetrad. Tetra means 4, so we kind of have 1, 2, 3, 4 chromosomes lined up together. All right, and remember, these code for the same genes, but you have mom's version of the gene and dad's version of the gene, right? So maybe we've got the gene for eye color here, the gene for hair color, whatever the gene happens to be. You have mom's version and dad's version. So what happens in this tetrad is something called crossing over. So during crossing over, segments of non-sister chromatids, right? So sister chromatids are the ones on the same chromosome, right? Those are exact copies of one another. Non-sister chromatids would mean one from one chromosome, one from another chromosome, break and reattach to the other chromatid. So this creates genetic diversity. So let's see how that's happening here. So they kind of like wrap around each other super tight, give each other a really big hug, and then when they open back up, they've exchanged some genes. So we can see here, some of mom's genes ended up on dad's chromosome, some of dad's genes ended up on mom's chromosome. So this is one way that we get some shuffling of genes that helps to lead to genetic diversity, which is a good thing for organisms. We want genetically diverse offspring. Okay, so that's very important. Looks something like this. So we've got the chromosomes coming together and they end up switching some pieces. So then next we've got metaphase 1, in which those tetrads line up again across the center of the cell. So with metaphase in mitosis, right, the chromosomes lined up, now the tetrads are lining up. Okay, because the first round of meiosis, metaphase 1, the homologous chromosomes are going to separate, not the sister chromatids. That happens later. So the homologous chromosomes are going to separate. This is called independent assortment, which means the position that they line up in is random. It happens randomly, different every time. So this leads to even more genetic variation. So sometimes mom's chromosome's on the left, dad's is on the right. Sometimes it's vice versa. Okay, so the arrangement of these chromosomes is important because it's random, and this is called independent assortment. So this is the second thing that leads to genetic diversity in meiosis. Okay, so we see they could line up like this, or they could line up like this. Again, it's random. Anaphase, the homologous chromosomes separate to different sides of the cell. The sister chromatids still remained attached, so we still have X's here. The chromosomes still look like X's. And then telophase, essentially we're separating, right? Each pole now has a haploid set of chromosomes, so half the number of the original cell. Cytokinesis occurs and we end up with 2 cells now. So where we started with 1 cell with 4 chromosomes, now we've got 2 cells with 2 chromosomes. Then the second phase of meiosis happens. And this is very similar to mitosis, essentially. Same as mitosis, nothing else happens. DNA doesn't replicate because we already have X's here. Metaphase 2, line up just like mitosis. Line up across the center of the cell. Anaphase 2, same thing as mitosis. The sister chromatids separate to opposite sides of the cell. Telophase 2, same. Okay, so the new nuclear membrane are forming around each set of chromosomes. But now the end result, since this happened twice, we end up with 4 cells. So 4 haploid gametes. Haploid, they have half the number of chromosomes as the original cell. 4 haploid gametes are produced. So we got 4 sperm or 4 eggs. And you can see here that they're each genetically different, right? This one has one dark chromosome, 1 light chromosome. This one has 2 light chromosomes, 2 dark chromosomes, 1 light, 1 dark, right? So this is showing how those genes get shuffled up during this process, which is why one individual could have several offspring, but they all look different, right? And function a little bit differently. Some may look more similar than others. Some may look very different. It's because of this amazing shuffling of genes that happens every single time. So every single sperm, every single egg is genetically unique. So to sum up this amazing process, meiosis makes gametes, or sex cells, which have a haploid number of chromosomes. Half the number of chromosomes of all our other cells. 2 gametes come together during the process of fertilization to create a diploid zygote. Lots of new words here. We're going to practice. It's going to be okay. Crossing over and independent assortment of homologous chromosomes during metaphase 1 lead to genetic diversity in gametes, therefore the offspring. So again, these 2 really important processes, that crossing over where the homologous chromosomes hugged each other super tight and switched some genes. That's one thing that leads to genetic diversity. And the independent assortment, the way that the homologous chromosomes line up in metaphase 1, right? It doesn't matter what side they're on, they switch every time. So that also leads to genetic diversity in the offspring. Okay, so this causes every single sperm, every single egg to be different. All right, I know that was a lot. If you need to go back and rewatch it, go for it. Let me know if you have questions, and I'll see you in class.